Hi there. Today we are joined by Dr. Christopher Armstrong, who is the director of the Melbourne MECFS Collaboration. It's so great to be joined by you today, Dr. Armstrong.
Thank you very much, Rebecca. And thanks to OMF to kind of be doing these interviews and supporting us. We're very grateful for what we're able to do.
Of course, we're so excited to have you. And today we just really want to talk about the updates and the progress that's going on at the collaboration. So I wanted to start off talking about something exciting that you're working on, and it's called the Ocular Motor project. So this is being conducted in collaboration with Associate Professor Joe Fielding and the team at Monash University in Australia. And this project uses high-performance cameras to monitor eye behavior in MECFS patients. And I know this has been used to develop specific signatures for many other neurological diseases. So with this ocular motor study specifically, you're seeking to categorize eye movement changes in MECFS with the goal of identifying an ocular motor signature that is unique to this disorder. So I'm wondering if you could please just discuss more about this, the process, and why eye movements.
Right, yeah, that's a great place to start. So this is a collaborative project, as you mentioned, with the group at Monash, led by Joe Fielding. And so they've been doing this type of research for many years. This is obviously their specialty, and they've been applying this to a number of different neurological diseases. And so the concept behind it is that the process of following things with your eyes, sensing where they move, having the motor and proprioception parts of your brain kind of working to move the muscles in the eye to then follow it. And even if it does kind of some other tricky things like flashes on and off and all that sort of stuff, like the anticipation, all these different things are just constantly requiring sensory and motor control, and many different parts of the brain. In fact, over 50% of the brain is kind of lit up when they're doing these type of activities. And they are simple activities, but it's actually a very complex system if you really think about how amazing the eye is and what it's able to do. And so through this type of process, when you're actually doing these different eye movements, different areas of the brain are kind of being lit up depending on how you test them. So a very simple test is to simply have a dot in front of the participants, and that dot may disappear and then reappear 5 centimeters or 10 centimeters to the left and the right. And that will come at random effectively. And so the participants will be looking at that dot at random when it disappears and reappears somewhere within that 5-10 centimeters to the left or 5-10 centimeters to the right. And so what we can determine is kind of their reaction speeds, how the velocity of their eye movement, the acceleration, deceleration of those muscles. And so that's a very simple task. They get quite a bit more complicated from that. A more complicated version would be asking them to look to the opposite side to where the dot is, because there is obviously kind of involuntary type movement where you want to follow where a dot or something goes. And so if you're looking to the other side, that requires a little bit more of cognitive processing. And so if you start to see like different patterns, so they're okay with following the dot, but then they seem to struggle when there's a little bit more complication, you can start to work out which particular problems or signatures that may be specific to people within that illness. And so that's kind of like a brief kind of discussion of how it's valuable. I think what we're trying to do is effectively monitor this to see if we can develop a specific signature for MECFS, or even the fatigue phenotype or post-exertion malaise as well. So we're trying to target it all. And the way that we're trying to target that is we're doing multiple different tests, and it only takes like 10-15 minutes at a time. So we'll do 10-15 minutes on a day, and then we will have effectively a second day we'll follow up where we come and do another test. And so that process is trying to determine what's kind of happening during the test on the day, but then also is that test creating and it's very cognitively exhausting. It's actually requires a bit of energy, and to see if then the next day if there's an improvement on that type of test versus not improving. Usually in healthy people you would get an improvement the next day if you're following up a test like that. And so what we're doing, we're comparing between healthy and MECFS for this test itself, just to see whether we can develop this signature. And then from there, it will be in comparison to a lot of the other signatures that they've developed for other illnesses as well. And so this to us is an exciting process because it's a very quick, simple thing that can be done. They are developing headsets that should be able to utilize these tests within specialist doctors' offices. So we are targeting for that future. It doesn't happen at the moment, but it's gonna be happening in future in some capacity. And so we're targeting for that, so that when we have this ocular motor signature, we can get an objective measure of perhaps either MECFS or sort of specific symptoms and their severity at different points.
Thanks so much for explaining that a little bit more. I know that's exciting news for people with MECFS, that we can find a way to hopefully have more insight into diagnostics without having an invasive procedure being done, because as you mentioned, it's something that's in office. It's not, it's not as comprehensive as, say, like a lumbar puncture or something like this. And I know eye movement, and also in MECFS patients specifically, so many talk about symptoms such as slow tracking eye movement.
Yeah.
And this is why a lot of patients specifically are resonating with this study, and it's coming out and they're saying, I've seen a lot of people mentioning, oh, well, I resonate with this because I have slow tracking eye movements, or I often feel, especially in a crash state, that it affects my vision.
Yeah, I mean, when I first looked at this this type of technology, I was kind of like, this is like a perfect hand in glove fit for MECFS, especially because of some of these extra eye component symptoms that patients report on top of the neurological aspects to this. And it's the simplicity. It is still an objective measure. You can't alter the speed at which you look at things, and you may not even realize you can't. I mean, slow tracking, you can probably, maybe slowly try, but your eyes really moving in jolts, and these like high performance cameras are really picking these up, the acceleration, the stopping, but then also the blinking and all the type of different aspects of the eye and their movement as well. So there's a lot of data here. It's huge amounts of data. We pull very simple measures at the moment, but we will also be getting statisticians to do AI-based analysis of the very raw data files, which is like the video files, to pick up a lot more of the sensitive information that might be in there. So there's a lot to do with this type of project. Follow-up projects in future, we'll be looking at MRI in conjunction with this to then determine the areas where there might be quirks in relation to the signature. And then you can determine, while doing the test, while they're in the MRI, to see which areas are kind of lighting up during different aspects of this. So there's a bright future for this type of research, I think.
That's quite fascinating, and I do think it's going to be important moving forward. So thanks again for kind of describing that more for us. And something else that you're focusing on a lot with this patient population is something called metabolomics. I'm wondering if you could please first explain that a little bit for people that might be new. What is that? And then can you just tell us more about this quick and inexpensive method for metabolomics? It's an at-home sample collection process that you are developing.
Right. So that's multi-stage questions. There's a few different things I will talk about, but initially the introduction of metabolomics is really the measure of metabolites, and metabolites are things like amino acids, sugars, all small kind of compounds that you may see in different energy drinks or isotonic compounds that you see. So there are lots of different things that count as metabolites, but really they're the smallest breakdown of larger products that we know, like fats and proteins and sugars and all that sort of stuff. So all the things that we eat are effectively broken down into these, and then they go into the body and they're utilized for full biological processes that cells are. So cells are effectively machinery that move around metabolites, and they do that in a way that produces energy. And so what they're trying to do is move metabolites to produce energy, but they also utilize metabolites to produce more machinery. So they're like, it's pretty much like the fuel that allows cells to do everything that is important. And so looking at metabolomics is trying to look at the ratio of how these compounds are existing in combination with each other, but also compared to different populations. So we might look at the concentrations or the amount of these metabolites in a biofluid like blood or urine, and compare that between someone with a disease like MECFS versus someone who is healthy. And so that's a really basic way that a metabolome experiment might be set up. We're actually really interested in the longitudinal type design, where it's the individual person taking samples over the course of days, weeks, months, and taking multiple different samples over that period of time to see how they're really changing. And the value of doing that is because metabolites are impacted literally by everything within you. Like there'll be the fact that you're maybe sitting down and having some cereal or going for a walk, it's going to be changing the way that these ratios of metabolites kind of are existing within your blood or in your urine, depending on what you did. And so this is why it's a really great tool to looking at the dynamics. And it's also the closest field of measures that is related to the phenotype, like symptoms or how you feel. Obviously, taking a walk is a type of experience, and so it's going to be impacted by everything. And so that's why it's so very sensitive, but also gives you a lot of information and detail about fluctuations, which people with MECFS would understand the dynamic nature of the illness, the way it responds to different stresses externally, but also the way it can just change without them really realizing with the symptoms and severities. And so we're kind of impacted by this all the time. And so what I think, what I would talk about the inexpensive method. So we use a machinery called NMR, nuclear magnetic resonance spectroscopy. You don't need to know that in detail, and I won't explain that. But there's two different types of machinery that kind of utilize, that we can measure metabolites with. We measure a whole lot of them. And the one that we specifically use, it measures a smaller number of metabolites, but it can do it very robustly, very quantitative, very accurately, and it does it very low cost. And we're trying to make our system more low cost than it already is. So currently running a blood sample to get a panel of metabolites measured through our system would be something like, or at least in the past, it would be something like $15, $20. And then now it's we're trying to get it to a point where it's maybe $1 or more, just over $1. And so what that enables us to do is really take a lot of different types of measurements. And so the at-home system that we're actually developing, which is actually quite a novel thing, this hasn't been utilized in any other disease, is we like to use these wearable tracking devices. So they're taking heart rate variability, things like Fitbit that people might be familiar with. We have those, but also we give them collection kits so they can collect urine at home. We can also collect blood, but we're collecting urine, and they can collect all the urine samples they have during a day, for instance. And so, yeah, what's really fascinating about that is that urine itself is excreting all the things that were within the body, in the blood, or not all of them, but most of the things that were in the blood that were kind of excessive or not necessary. And so, you build up an excess over the day. And so if you capture all the urine within a day, you're capturing effectively all the things that are within excess in that period of time. But you're also capturing things like diuresis events, which are kind of mass exodus of the blood, pumping metabolites into the urine. And so what we have is, you might have people who will pass urine 7 to 10 times within a day. And so we can collect 7 to 10 urine samples from an individual in a day while they're getting tracked with the continuous tracker event. So we can, if we date and time it, we can kind of go back and see kind of like where, which portions they actually collected and what was happening during that period of time, which we can relate to maybe the changes that happened during that period of time compared to the portion before. So every time, it's almost like a timestamp of everything that you've done during the day. So we're kind of taking advantage of this thing, and we think it's really, really intriguing because typically, people would sample blood, and they would sample blood in combination with these wearable devices. The blood is fantastic, but the one issue with it is whenever you sample it, it's really just at that moment of what blood looked like. It could change the next second or the next minute in some capacity. And so you're not going to capture that just by sampling it. And it's also a bit more invasive. Obviously, urine is not invasive of a collection. So yeah, we think, and completely painless. So urine collection is, I think, something that we're really excited about. And there are a lot of things you can kind of do when you, especially when you're measuring just from the same person, which we're really interested in the dynamic changes over time.
And I think that's also, I wonder if that's also beneficial for sometimes with MECFS, they can actually these patients can't get to the lab easily, especially if they're homebound. So this urine collection over time might also be beneficial for these patients who are homebound. It lets them kind of track hour by hour, however many times, and it gives us this really in-depth picture without them having to necessarily leave their house for this.
Exactly. These are the benefits, and we are trying to do a lot of at-home work. So if we do try and ask for blood collections for longitudinal studies, they're using like finger prick devices where you're collecting a little bit of blood, but urine is so much more quantity. We can do a lot more with it, and it's non-invasive, and it's really cheap with this system. So the system, because it's $1 per sample, or we'll be getting down to something like that in terms of a rapid profiling, high throughput profiling. Because of that system itself, we're going to be able to measure all these different compounds, but it does require a bit more fluid than you would get through blood pricks. Like it would require a decent, probably an official blood collection with a tube that you would do at a pathology lab. So that's not really something that we would like to do with patients, getting them in to take full bloods all the time as much as we want. So if we give these kits to patients at home, it's pretty straightforward, and you don't have to collect all your urine. The whole idea is there's a collection jug that kind of sits within within your toilet, and then you can give a sample, and then it will collect all of the sample, and then you can it has like a measuring cylinder thing, so you can effectively just tell us how much, and you can take we take a small sample, we provide you like a little drop, these little lab type dropper things, and you just take a small sample into a small tube, and you can put it in a storage. We have a storage container that can sit in your freezer or something like that. That's multiple contained, so you won't get any contamination or anything like that. And they can probably, they could collect hundreds of samples alone, and it won't cost us that much for us to run everything. And so we get a really detailed resolution of information. It just really depends up to them of how much they would want to collect and how much they want to detail. And so things that we're excited for the use of this type of style of project would be for tracking over the course of a crash. Like if we give the samples to a patient and then they effectively wait until they feel like they're going to have a PM event, and then they just start sampling that whole period if they can. Yeah, and so this would be the only time we'd actually be able to capture every moment of that system up until even recovery. And it really comes down, it's really limited by how much the patients are really willing to go through, do this for every sample and their setup, but in the help that they have. But that's something we're excited about exploring, for sure.
Absolutely, that does sound very exciting. Cost effective is always good. So
with
especially MECFS.
When you want resolution of samples, when you want hundreds of samples from one person, you have to think about it for cost effectiveness, unfortunately, because it's
absolutely.
Otherwise,
a lot.
Yeah, well, like usual
hundreds.
Other methods would cost like $100 a sample, and so you start to thinking about okay, 200 samples, that's $20,000, right, for this versus $200. So the value adds up a lot, and so this can be combined into a system that may be affordable for monitoring and tracking at some point as well, beyond just research.
It's exciting.
Yeah.
Something else that I'm excited about that I'd love to have you explain for us a little bit is your pediatric study. So one of your areas of focus is MECFS patients that are actually ranging in ages from 12 or 13 to 18 years of age. So why is it important to look at this age group specifically? And then can you also just explain a little bit more about this study and how it is done?
Great. Well, yeah, I mean, for one, very little biological research has been conducted on pediatric patients with MECFS. And they do have a slightly tweaked definition. In fact, their definition requires them to have the illness for 3 months. So you can collect them earlier in their illness. They have a lot less comorbidities. As I said, they are earlier in their illness generally. They mostly also have a very similar trigger, EBV virus, which is infectious mononucleosis, or glandular fever, depending which country you're in. And yeah, so these are like advantages of looking at this type of patient group for understanding MECFS generally. Is the actual patient group itself, but also we want to see what differences there might be within pediatric cases. And it's obviously a really important time. And it's a really, it's never a good time to get MECFS, but during its developing years, it can really derail lives entirely. And it's something that probably needs more research into it, since there hasn't been really any. So we are really doing a large scale, in-depth, because of that, trying to make up for the fact that there has been such little research in this area. We're really doing the works of different types of things to explore as much data about these patients as possible. And so that looks like the patients come in, they give a blood sample, they have a number of tests, cognition tests, and a number of different surveys that they file out, and then they'll go through an MRI.
Wow.
And that will be, and then they'll be given a kit, a take-home kit, and that take-home kit is to do collections during an average day. So they'll have maybe 5 or 6 collections during an average day. 6 or 7 collections during an average day, and then 5 or 6 collections during post-exertion malaise events, for both a blood prick but also urine collections, similar to what I was just describing before. And so that'll just be for the day, for when they measure that. And so the way we get an average day is we tell them when to collect it, regardless of potentially how they feel. If they feel too bad to collect it, then they obviously can, choose a different day. But we try and make it so that they are doing it on the days that we set, so that we can get an idea of what average looks like. And so that'll be different fluctuations in symptoms. So there'll be symptom information at the time as well. So we'll have different perturbations of symptoms over a period of time. And we'll be looking at all the metabolites, proteins, what we call transcripts, which are effectively when genes are kind of signaling what proteins to be made. And those are the cells, so signaling what proteins and enzymes to make. So we see those signals, and we're going to be looking at whole genome sequencing, so their entire genome, and potential perturbations that may be in there that may explain some of the issues that we are seeing in all the other measurement profiles. And then also the blood sample that we collect on the first day will be a large scale blood sample, and the collections that they do at home are just very small pinpricks. So we can do very limited studies on the pinpricks. We will look at cytokines. We'll probably try and look at autoantibodies, metabolites, proteins. But on the initial blood sample, with the big sample, we're able to get cells, collect cells, and we'll be doing some cellular-based metabolism assays to assess the cell's ability to make ATP, or at least the way that they use metabolites in the production of energy, I should say. It's a more simpler way. And so that's the combination of things that we're doing on these participants. And so it's really kind of coming together with a whole lot of different types of research.
So important to be looking at pediatric MECFS. I think it's not talked about enough, perhaps, and especially now with incidents of COVID, so many kids that are getting these symptoms, long COVID, they're now qualifying for MECFS diagnosis. So likely the work you're doing is even more urgent now than ever with all of these kids that are getting sick.
Yeah, definitely. we want to make sure, obviously, that we're capturing or looking at everything. And so it's just it's a benefit to them to see whether we can see it, but it's also a benefit to MECFS, and their participation in these studies is obviously extremely, we're very grateful for that, and it's extremely valuable for the research generally, and MECFS generally. I mean, these patients do have MECFS. They often will continue to have it for their life, or for a long period of time at least. So it's something that we want to capture early and get as much information as possible.
Absolutely. So something else that you're studying in depth, I know we talked about this a little bit, but it's post-exertion malaise flares, PEM, and you're studying this in MECFS patients, but also in other groups such as post-treatment Lyme disease syndrome patients, long COVID patients. And what you're doing is you're looking to identify biological patterns that correspond to MECFS or post-treatment Lyme disease or long COVID symptoms. So I'm wondering if you could just tell me a little bit more about this project.
Yeah, I could definitely do that. So this is in collaboration with Stanford, with Mike Snyder, actually, who's professor there. It's under his lab. So this is something that they're kind of all conducting there. It's called the Crash Course. And so, yeah, I'm very happy to be involved in this project. They have a great team working on this. It's effectively, as you described, it's looking, and kind of similar setup to what we're doing with longitudinal for pediatrics. It's looking at 7 MECFS patients, we're looking at 50 MECFS patients, or 50 to 100, and we're looking at 7 days every 2 weeks. So 7 time points every 2 weeks. So it's over 14 weeks that we're kind of doing this study. And during that period of time, they're taking effectively these little blood samples through a device that kind of attaches to the arm. It's pretty painless in terms of collecting that blood. And then we're also using that device to collect those samples during periods when they have post-exertion malaise. So there'll be 6 samples taken during that period. And then there's going to be 3 samples taken when they're having very good days, so relatively good days for an MECFS patient. So it doesn't have to be any more defined than what they would deem something as a very good day for them, based on their disease and how they've been during that. And so the idea is to try and get a baseline again over the every 2 weeks, where we tell them when to collect, and then again with getting post-exertion malaise samples. And so it's also what's kind of beautiful about this project. I mean, it's working with the Snyder Lab, and they're the kind of the inventors of this type of research. So they began quite a long time ago, and so they're very well adept at doing all this. And so we collaborate them with a lot of our longitudinal-based projects because of their expertise in this. And so that's one beautiful part about this, but it's also a combination of looking at MECFS with also post-treatment Lyme disease or chronic Lyme patients, depending on how they like to be reported, and then also looking at long COVID patients as well. So in combination, trying to see, we're trying to look at within the patients how different profiles compare, over time, looking at their biological changes as reflected by their symptoms that they report, but also they will be wearing wearables like Fitbits and getting all that type of information as well. And so we're looking at within each patient, within each group, but then between the patients within the groups, and then between the groups effectively as well. So looking to see how much similarities and differences there are, whether there are subtype components in this, or whether there are, it's really a lot of the same process. It'd be very interesting to see if there'll be some patients with MECFS who look like the same as people with long COVID, and more similar to those maybe with something long COVID versus other patients are diagnosed with MECFS, whether that happens. And that could very well happen. So we're really looking forward to seeing that data when it comes out. It will be very good at confirming potential subtypes across different illnesses, or different variations of what we would call multisystemic complex chronic diseases. Yeah.
That's so important. I think so many patients, MECFS, any of these chronic complex diseases, they're not one size fits all. So people vary so much in their symptoms, and their post-exertion malaise may present differently. So I think it'll be really interesting to have a in-depth look at what exactly is happening with post-exertion malaise on a molecular level.
Yeah, for sure. I think definitely it's the defining feature, and these other diseases, long COVID, post-treatment Lyme disease, they have, you know, flare events as well, and so it may be quite different in the way it behaves, and so we really want to see how there might be differences as well. And that's another point, like if we get flare events, what we call a flare event or a post-exertion malaise, or just a rampant increase in symptoms and different symptom varieties kind of popping up post some type of stressor component, then we have, we want to see whether there are similarities, whether there are differences, and better kind of characterize what they look like, and maybe work out signatures that may separate them biologically, if there are things that we can pick up within the blood. So their system there, it looks at very small amounts of blood. It's pretty critical. They're able to look at, you know, thousands or about 1,500 different biological markers, which is including, you know, proteins, lipids, metabolites, cytokines, autoantibodies as well. So they're really on top of a lot of really interesting stuff there from this tiny amounts of blood. So we're excited to see how this kind of goes, and it's actually progressing now. So recruitment phase is in action.
This is all, the technology is getting better, and when we look at the things like the blood and urine in conjunction with wearables, wearable technology is getting to be very important in precision medicine. So we're able to understand people more on a personal level through these technologies. So we will be really excited to hear more updates from all of these studies that are going on in your neck of the woods, because you are doing so much important work right now. And right now it is our May Momentum campaign. So we ask you, if you are able, please consider supporting the efforts of directors such as Dr. Armstrong, who's working so hard at the Melbourne MECFS collaboration. So thank you again for joining today, Dr. Armstrong.
Thank you very much, Rebecca, and I appreciate you taking the time to interview me, and thank you again to Open Medicine Foundation for their support, and to the community itself for the support that they bring to Open Medicine Foundation. Obviously this type of work doesn't get done without that help. So
that's right. That's absolutely right. It's a community effort. So thank you to all.